I'm Shunya Sugita of OA Laboratory, Department of Computer Science, School of Computing, Tokyo Institute of Technology in Japan. Today, I'd like to talk about drug target affinity prediction using applicability domain based on data density. One of the important steps in drug discovery is to predict the binding affinity of drug target pairs. In such cases, computer is helpful, but the compound and target space is so large that predictions by machine learning are not always valid. Therefore, we propose a method to indicate whether a prediction is reasonable or not, depending on the applicability domain on data density. Experiments show that the method with KNN works well. The accuracy of samples for which AD by KNN was classified to out AD was mostly poor, while the accuracy of samples classified to in AD was mostly good. It costs $2.6 billion to develop a new drug, and it takes an average of about 12 years from development to approval and market launch. Therefore, reducing the cost of drug development through computation has become an important issue. Many drugs effects by interacting with proteins. For this reason, computational predictions of the affinity of drug target pairs to interact and how likely they are to interact is important because it reduces the cost of experiments. In particular, as shown in the figure below, prediction of drug target affinity by chemogenomics, which predicts a non-affinity from non-affinity by integrating information on multiple compounds and multiple targets, is relatively fast and can predict a wide range of affinity. However, machine learning does not always make reliable prediction. As shown in the figure below, the error between the test data and the prediction curve is large in the red domain, with less training data. In particular, chemical spaces such as compound space and target space are large. Therefore, we use the applicability domain for drug target affinity prediction. A machine learning model can perform reasonably well in the applicability domain. The applicability domain is estimated by quantifying the distance from the model to the data. There are several ways to quantify this using training data and prediction models. Depending on the applicability domain, we can classify data with low reliability of prediction and data with high reliability of prediction. In general, the prediction error for data that classify to the out AD is so large that either no prediction is made or the prediction result is ignored. In this study, we propose a method for predicting drug target affinity that considers the applicability domain based on data density. As a method to determine the AD, we apply the k-nearest neighborhood method and the one-class support vector machine method. Both methods use the closeness from the test sample to the training data as a criterion to determine whether the data is inside or outside the AD. In the KNN method, the distance between a test sample and the model is average of the distance of k training data near the test sample. If the distance is within a certain range, it is classified as inside the AD, and if it is greater than a certain range, it is classified as outside the AD. In this experiment, we set k to 5. The other approach of CSVM is a kernel-based classification method that runs a discriminative boundary for the AD by the training data. Both methods use the data density of the training data. Two figure are simple illustrations of the AD by KNN and OCSVM. Two types of features are used. The x-axis shows the first features and the y-axis shows the second feature. Blue is training data. Green is the test data inside the AD, that is the test data outside the AD. The orange area is the AD. You can see that the area with a lot of training data is painted in orange. In both methods, features are normalized and then weighted by multiplying the feature importance calculated by the machine learning model. In addition, nu is a parameter that determines the size of the AD. The smaller nu is the wider the AD becomes, and the larger nu is the narrower the AD becomes. For these experiments, we use the SynBoost model. The implementation used the publicly available code. The features were also the same as SynBoost, and 98-dimensional features vector were generated for each drug target pair sample. In the evaluation experiment, 3 data sets are used. The affinities are recorded as kinase dissociation constants, kinase inhibition constants, or KIB scores. RMC is used as the evaluation index. For this, the parameter nu, which determines size of the applicability domain, is varied in 4 ways. We have conducted evaluation experiments with the 3 datasets shown in the table, and we will introduce 2 of them here. In the table, the Davis dataset is a dataset of 68 drugs and 442 targets, and 100% of the density of the dataset means that all of these pairs, 442 multiplied by 68 affinity values, are known. On the other hand, about 3/4 the KIBA dataset is unknown. In the proposed method, training data is used to train SynBoost and to determine the boundaries of the applicability domain. Using the learned boundaries of the AD, the test data is first classified into in-AD and out-AD, and then the SynBoost model, which is also trained on the training data, is used to calculate the prediction results for the entire in-AD and out-AD test data. The figure below shows the results of each method on Davis dataset. The baseline is the accuracy of the entire test data is drawn to the left. When the KNN method has the narrowest AD, the RMC of in-AD is the most improved. Also, the RMC of out-AD is the best when the AD of KNN is the widest. In other words, we can see that particularly inaccurate classification is possible when the AD is widest. On the other hand, OCS-VM does not classify inaccurate ones as out-AD, unlike KNN. Next, we show the result for KIBA dataset in the same way. As in the Davis dataset, the RMC of in-AD is improved the most when the AD is the narrowest. Also, in KNN, the RMC of out-AD is the largest when the AD is the widest, nu equals 0.02, and inaccurate ones can be classified as out-AD. OCS-VM is able to classify inaccurate data as out-AD better than the Davis dataset. This may be due to the fact that the KIBA dataset has a lower density of dataset, and the AD can be set up better for datasets with lower density. Using the KNN method, we examine the change in RMC of in-AD and out-AD for the Davis dataset as nu was further varied. As a result, the RMC of in-AD tended to improve as the AD became narrower. When the AD is the narrowest, it's a pretty good prediction. In other words, the distance from the model as quantified by the KNN method tends to be more accurate about RMC the closer this distance is. K is a parameter in KNN and we also examine the accuracy when moving this parameter. This table shows all the value RMSE of at AD. So the higher is better. We can see here that k equal 1 is good for classification. So it is better to focus only on samples that are closer to each other. K equal 1 to 5 is appropriate because increasing k to 10 or 100 does not improve the RMSE of in AD. Since Davis data set is dense, k equal 1 is better. But depending on the density of the data set, the more sparse it is, the larger k should be. Regarding the accuracy of the present method, the accuracy of the Shin-Boost model is good. So the accuracy of the proposal method was almost the same. This may be because the accuracy of Shin-Boost is not only simply good, but also because the Davis and KIBA datasets are dense, and especially the Davis dataset has non-binding affinity for all pairs of emerging drugs and targets. Therefore, it may be appropriate to classify the entire test data as in AD. If we use a more sparse dataset, the AD by data density may work more effectively. As for the setting of the parameter new, since the KNN method improves the RMSE as new increases, we cannot choose new such that the RMSE is minimized. Therefore, we can use the distance calculated by this method as the reliability index of the prediction. In this case, the parameter nu is no longer necessary. Conclusion, due to the large compound space and target space, we propose this method for reliable drug target affinity prediction, considering the applicability domain by data density. Evaluation experiments showed that improvement in accuracy with proposed method. However, there are still some issues regarding the accuracy and the setting of the parameter nu. Acknowledged and shown in the slide. Thank you.